Yeah, I think everyone know about yesterday we have discussed about recombinant DNA technology, and before that again I will repeat, I will remind you all. Uh, yeah, please be sure with the new pricing system. Okay. Try to pay within 15 days. Okay. Mm. Then yesterday we have discussed about recombinant DNA technology. What else? What else we have discussed? What are the points we have discussed? Can you summarize anyone? Dublina? Um,
Sorry, sir. Yesterday I didn't attend the class and
okay. Nitish?
There's steps in recombinant DNA technology or
Yes.
certain tools of recombinant DNA technology, then techniques, and then uh, types of restriction enzymes, and then cloning vector, and
Yeah.
then sir about some dolly seep.
At TI
Just I
platform.
have given hint. Yeah. Any other steps, tools, techniques, types of restriction enzymes, and what are the different cloning strategy vectors, and a little bit about some extra points we have discussed. And we have discussed the transformation methods. Okay. We'll again briefly I will explain in detail. Okay. There are different types of restriction enzymes. Yesterday we have discussed type 1, type 2, type 3. Type 1, which means that recognize the specific DNA sequence that called as your restriction sites, but cut at random site far from the recognition site. Like it will introduce the break from the recognition sequence. You know what is the recognition sequence? Or like what is the character of recognition sequence? If some sequence is said to be recognition sequence, what are the main characters?
Isomeric repeats.
Yeah. Okay. If you read from one this direction, you can read from this direction. See, example Liril. You take LIRIL, LIRIL. Okay, there is again different types. I'm not going detail. Like these type of sequence present on different strand or either the same thing, same position or the same strand. There are different types, but you just remember if you repeat from both sides, it will be read as same. Okay, that is called as recognition site. There are different recognition enzymes. Examples are there. Okay, for each section, there will be different examples. For type 1, echo KI1, like 1 is a strain number. Okay. And you should also remember what is the how they will nomenclature of the restriction engines. Okay. What is the strain name? What is the different what is Roman letter? Why it is capital? So everything you should remember. Actually it is not it's not been capital. Maybe I wrongly written. 1 minute. Yeah. And there is a type 2 restriction enzyme which recognize this palindromic sequence, that is recognition only, and cut within the or near the recognition sites means it is so far, means it is either actually this word also wrong, it's exactly within. Okay, then type we have several examples like core I1, hint 3, BM. BAM-H1. There are several, many other. Nearly, you can check the like a restriction site library. I think in website there will be a lot of restriction enzymes every day. They will be discovered, and it's used in the recombinant DNA technology. And when it comes to type 3, recognize a specific DNA sequence, but either distant downstream from the your palindromic sequence or your recognition sequence. Okay, it is, you can see, if you want to cut at specific site, but they are cutting the DNA sequence at different sites. That's why this type 1 and type 2 they are not used in the recombinant DNA technology. There is again other classification called as isosesomers and neo-isosesomers. And yesterday I'm unable to pronounce this actually. Uh, that's why I kept the slides. Isosesomers and neo-isosesomers. Isosesomers are nothing but they are the enzymes. For example, you take SAM1 and XML1. These two enzymes having a same recognition sequence or same recognition sequence, and they cut at the same site. For example, they having ATGCTU as a recognition sequence. Means this both SAM1 and this XMA1, they will cut at this ATGTC restriction site and at the same position between T and G. Okay, here you take, here CCC, GGC is recognition sites. You take, for example, it is cut between C and G. It means both the enzymes will having same recognition site and they cut at the same basis, between the same basis. But there is another thing. For example, HAP2 and MSP1, they're having same recognition sets, but they cleave at different sets. For example, HAP1 cut it in between C and C, and MSP will cut it between G and G. This is the what is the
Okay,
difference between.
super.
Hello? What happened?
Sorry, sir. I mistakenly unmute some of my friends. Sorry, sir. Sorry
Okay,
for
okay,
this.
okay. Yeah, they are neo-isosomers. Okay? SPA1 and MSP1, they cut at they have same recognition sequence, but they cut at different sites within the recognition sequence. That is called as neo-isosomers. Then they are again there will be a different thing. There will be different classification. Blunt end cutters and sticky end cutters. Means some they produce blunt ends. Means they will produce blunt ends like this. And some of the enzymes they will produce overhangs. For example, there will be overhang like this. Is 3' overhang or 5' overhang. This is called as sticky ends. Some of the like Ecora and BAMH when they are sticky ends. They will create sticky ends. Then there is another terminology is called as star activity. It means that, for example, Ecora take at some condition it has to cut at some recognition site, but instead due to some other external condition, it will not cut at that site. Means under suboptimal conditions, some restriction enzymes cleave at non-specific site. This is called as star activity. Star activity is desirable or not desirable in recombinant DNA technology.
Non-disabled, sir.
Yeah, because the cleave at non-specific site means that everything will be disrupted. You will not get your specific sequence and you can't get your desirable clone. So that's why star activity is not desirable. You should know what is star activity, what is sticky ends, what is blunt ends, what is isosesomers, what is neo-isosesomers, and different classifications. It is must actually. And restriction enzymes, who discover restriction enzymes? Read it, okay? Smith and Smith et al. Having discovered this one, these restriction enzymes. Please read about the history of restriction enzymes. Okay, you just type in a Google, need like who are the scientists, when they awarded Nobel Prize, everything will come. Then I also discussed about some after restriction enzymes, we need to have some marker systems in the vector so that we can easily select the transformants and non-transformants. There are different types of marker. One is selectable markers and another one is scoreable markers. Why we need markers? They are used to identify and track the cell or organism that has successfully taken up the foreign DNA. For example, uh, you added this particular recombinant DNA into the host. Some are they're all cloning, but among this, which the bacteria having, which colony is having recombinant DNA and which having non-recombinant DNA. That we need to decide. But how you will select? Because everything is looks same. The colonies looks same. But something we need, some marker we need to identify which clone is recombinant and which clone having the non-recombinant DNA. That is called as the markers in the recombinant DNA technology. There are several types: selectable markers, scoreable markers. First, we will discuss about what is selectable markers. It means that these are the genes that allow identification of transformed cells by enabling them to survive under some selective condition. For example, take your antibiotics. There are different antibiotic resistant genes, and we can also use some herbicide resistant genes. For example, you take antibiotic resistant genes, NPT2, and this you should remember what is NPT2 and which is coding for which resistance, and AMPR gene, which it is code for which resistance. Okay, NPT2 is mainly for kanamycin. Many times it asked in the exam. NPT2 is mainly for kanamycin antibiotic resistance. Amplicin means like AMPAR means for the amplicin antibiotic. For example, in this is the agar media. You add some antibiotic like amplicin. For example, this site may having your amplicin resistant genes. Because of insertion of this DNA, it will make disruption of this amplicin resistant gene so that what are all the clones which are recombinant is they are unable to grow in amplicin. So that first you have to multiply the clone in tetracycline, then you take replica plating, and this replica plate is grown in amplicin. Which clone is able to grow in amplicin? They are not recombinant. Okay, which are not able to grow in replica plating, they are having your transgene, because where the resistant gene is disrupted by inserting your transgene. That's why how the selectable marker are very important in tracking the identification of transformants and non-transformants. Then herbicide list, for example, you have this bar gene will be there. If you introduce this bar, like if there is a bar gene, if you spray herbicide also, it will not going to die. For example, it having a phosphonithracin, like phosphonothracin. If this particular gene is having resistance for phosphonothracin, that plant which having or cells which having bar gene, they are very resistant to phosphonothracin. That's why based on that herbicide sensitivity, we can able to classify which are recombinant and which are non-recombinant. There are classical examples. See, the bar gene is a classical example, and this herbicide also very classical. Phosphonothracin and PAT gene means glufosinate. There are this is a different example for selectable markers. I hope you understood what is selectable markers. Any doubts regarding this?
Okay.
And going first because yesterday we have some discussion about it. Then another marker system is called as scoreable markers. Means scoreable markers are they are also called as reporter genes. There are means these genes that produce a visible or a measurable trait that identifies the transformed cell without killing a non-transformed cells. Means we can do implant identification. Here, when you apply antibiotics, some cells are we going to kill, but here it will not happen. In the living cell, without any killing any cells, we can able to track or identify which is recombinant, which is transformed, which is non-transformed. This is the one of the greatest advantage of the scoreable marker over the selectable markers. Then there are different examples. You should remember because for the scoreable markers, beta glucuronidase and clean fluorescent protein, luciferase, and anthocyanin producing genes. And mostly used is GFP and GUS and LUC. Okay, luciferase, green fluorescent protein, and beta glucuronidase. What is the function? For example, this beta glucuronidase, if the gene is present, it converts X-glucan medium to the blue color. If the gene is not there, it will not convert the X-glucan medium to the blue color. By based on that, the color of the media after your cloning, you can able to decide that which is recombinant and which is non-recombinant. This is one of the great examples. Like advantage of this one. Without killing, it will not kill any cells. It just produces some color. We can able to score that. There is another marker system also, blue-white screening, that I will deal in detail from next slide. Then another marker system is GFP protein. Okay, whenever the cell which having, say this is the GFP. Okay, whenever the other cells don't have this GFP, but this having the GFP. Whenever it will exposes to UV light, it will reduce it will emit the illuminescence, green color illuminescence, fluorescence under UV light. Based on that, we can identify which is transformed, which is non-transformed, which is recombinant, non-recombinant. Okay, same way, the luciferase. It produces the bioluminescence in the presence of the luciferin. You should remember all the examples, like which is scoreable marker, which is selectable marker, and examples for different markers. For example, selectable markers, antibiotic resistant genes and herbicide tolerant resistant genes. For scoreable markers, beta glucuronidase and green fluorescent protein and luciferase. These were the different examples. Then when you see the difference between them, selectable markers and scoreable markers, they allow survival of the transformants. For example, you had to grow the bacteria in some media so that media, based on that, it allowing survival or killing of that particular colonies. Based on that, you are selecting your transformants and non-transformants. But it's just a visible, measurable trait. For example, color, fluorescence, like this is scoreable trait. Means we can able to, okay, this much color is there, okay, this much expression of the gene will be there. Like that. This is the one of the difference. Then allow positive selection through survival. Direct, it is not involving any positive and negative, just direct visualization. Then there are example like antibiotic resistant, herbicide resistant. These are GUS, GFP, and luciferase. And screening for survival, it is means just studying. For example, you are inserting a gene. Some genes are go in the leaves, some genes are in the flower, some genes are in the root. Where your gene is localized, where your transgene has expressing, can able to locate. Means for example, it expressing in a root, you just take the root sample and you visualize in the fluorescence. Sorry, in the UV light, you will get a fluorescence. Means okay, your GFP is here. Means your transgene also here. Means you can able to localize where your gene is present on the plant. That is one of the greatest advantage of the scoreable markers. Then, sorry, then another type is like another important marker system is blue-white screening by lag-Z gene. What is the function of lag-Z gene? Can anyone tell?
Beta galactosidase production.
Yeah, beta galactosidase. Okay. For example, which gene? For example, one is one fragment. There is, beta galactosidase is a polypeptide. Means it's having 4 polypeptide. One of produced by the gene, one of the produced by the another of is produced by plasmid. And there is a complete galactosidase, beta galactosidase, so that converts your agar X gal to agar X gal plus IPTs to blue color. If this gene is disrupted, it can able to produce blue color, agar medium into blue color. That is based on that we can able to screen. We can able to see this blue color one is recombinant or non-recombinant. Means you are having a lag jet gene. You are inserting some transgene in the place of lag jet. Means you are inactivating this lag jet by inserting your transgene. You are inserting a transgene in the region of the lag jet so that the function of the lag jet will be disrupted. Then the ability of the plasmid to produce one of the your 2 polypeptides which are necessary for the beta galactosidase function, they are disrupted. Then here, what you will going to expect? Either white colonies are recombinant or blue colonies are recombinant, transformant or non-transformant.
White
White
colonies are
colony
recombinant.
Yeah, here written. Okay, white colonies are because here when the beta galactidase is in functional form, then only it can able to convert the agar media to the blue color. But we are disrupting this black jet. We are disrupting this black jet by introducing our transgene, so that this function of this transgene this will disrupted. It can able to unable to synthesize beta galactidase polypeptide. Then we're not going to get the color transformation. That's why all white colonies are here recombinant. This is called as insertion, like black jet blue white screening. Blue white screening of recombinant and non-recombinant. Okay. Then how you do selectable by using selectable marker gene? For example, you are inserting. For example, you take amplicin. Your one gene is there, amplicin resistant gene. And you are disrupting amplicin resistance. Okay, you are disrupting amplicin resistant gene by introducing some transgene. Then you have to grow in tetracycline media first. Actually, here they have disrupted tetracycline. We can take this example only. Here tetracycline gene is there. We are disrupting this tetracycline resistant gene by introducing your transgene, so that the function of tetracycline will get disrupted. First, if you grow this colony in amplicin media, all colonies will grow because amplicin gene is not disrupted, but only tetracycline is disrupted. It can able to grow in amplicin media, amplicin antibiotic media. So you are getting a good number of colonies. Then you just put a wooden block on this plate. All this colonial, some part of these colonies will attach exactly same position. Then you take this wooden block and put in a new Petri plate which having tetracycline medium. So that when you incubate this plate, here's only some colonies are produced in this tetracycline. These are recombinant or non-recombinant. This is last. These are recombinant or non-recombinant.
Recombinant.
How? It growing in
Non-recombinant,
tetracycline.
sir.
Yeah,
Non-recombinant because they are having
non-recombinant.
disrupted.
Then here we having a position because it's exact replica of this plate. See, this is the replica plate which I have grown in the. This is the extension of this plate. See, you have to locate the position where your non-recombinants are there. Means they are non-recombinants. Okay, these are non-recombinant. Where the position which is not colony is not growth, they are recombinant. Then you this position, this plate and this plate exact replica. Then you locate, okay, here this position, it is not grown in tetracycline, but it is grown here. Then you select this particular colony and you do again elute this colony and purify the DNA and use it. Use it for transformation. This is how we can use antibiotic genes as a selectable markers. Understood what is selectable marker and a scoreable marker system? How we can use
Yes,
that?
sir. Yes, sir. One doubt, sir.
Yeah.
No, in that replicon provided, they are also give amplicin resistance of the gene, sir.
Yeah, because maybe vector having two these genes.
That's right, sir.
See, this having, for example, you take amplicin resistance and tetracycline resistance. Both will be there. Somewhere tetracycline resistance will both will be there. See, TCR and amplicin. If this is disrupted, it unable to grow in the tetracycline medium. If this is disrupted, it's unable to grow in amplicin resistant medium. For example, you're treating SAL1 restriction enzyme or BAM-based restriction enzyme, E. coli restriction enzyme, it will disrupt the tetracycline gene. That's why it is unable to grow in the media containing tetracycline antibiotic. For example, you are using PST1 restriction enzyme. This PST1 site is present in the amplicin resistant gene. If you cut with PST resistant gene, this amplicin gene is disrupted so that it unable to grow in the media which is containing amplicin. Then exact opposite uh, you will going to get. Exact opposite system you will going to get. Okay. Now you understood?
Yes, sir. But they have, can you pass before slides? Look, previous. This like here amplicin resistant gene is also there and lag
This
Z
is the
gene is
tDNA.
there.
Okay. Type plasmid. This is type plasmid. I will explain that. Okay. How there are different things are there. Okay. What your doubt? Here no amplicin resistance genes. How we can select?
Not this slides.
Here this slide.
Okay.
See the vector will look like this. Okay, this is PBR 332. There are many modifications. There are different vectors. There are different plasmid vectors. You can read it from book. Okay, there are several modifications. Okay, there are puck vectors. Are there? Normally we use puck vectors. The puck 18. There are several examples for there. University of California. Like there are many examples for there. They have different modifications. It's
Okay,
not
sir.
like this is the only marker system. We can include anything. Okay, you can include promoter, terminator, or you can add any other antibiotic gene. You will modify the plasmid. It is like unlimited.
Okay.
There are nowadays they are using several many things are there. Okay, and this bacteria we have to order a competent host. You can't do normal. You can't use normal E. coli. There will be a competent E. coli, lambda. There are several things are there. The things are not necessary. I understood. What is your doubt? I'm not getting. What
No,
exactly
sir.
your doubt?
I got, sir. I got.
Yeah, yeah. Then, okay, you have used now selectable marker. Okay, you identify transformants. You are done cloning, and you have your recombinant DNA, and you have colony. Means your recombinant DNA is multiplied n number of times. Next step, you have to transfer into. It is an E. coli. Everything is now in E. coli. You have to transfer this into plant. How you will transfer? There are some vectors called as there are some vectors called as type plasmid. Okay, type plasmid vector system. This having a tDNA region which can able to integrate in the plant gene. How? We will see in this figure. Okay. And yesterday I was discussing what is the different regions present in the type plasmid. Type plasmid having tDNA region and virulence region, origin of replication, open catabolism gene, and left border and right border. And within the tDNA, there will be axin producing gene, cytokinin producing gene, and open producing gene. And these were they will responsible for synthesizing the hormones, which are helps in the development of the gall. That's why the type plasmid, if whenever it is integrated into the plant, it induces the crown gall disease. Okay? And Townsmith and scientists was identified this as the type plasmid can cause a crown gall disease. There are several history was there. Please read it. Okay. Then how these left border and right border they are very important for the excision of this tDNA region. How we will see? We will, like how we will work? We will see. Okay. For example, this type plasmid, they release virulence genes. These virulence genes, there are different virulence genes called as Vir A, Vir G, Vir D2, Vir D1, Vir D2, Vir C, Vir E2, Vir E1. All are responsible, helpful in the excision of your tDNA from type plasmid and integration in the plant gene. How? For example, whenever the plant tissues, they will secrete, especially dicots, they will secrete some acetosyringe, like acetosyringine only. Okay, then like some phenolic compounds, that is acetosyringine, phenolic compounds, plant root exudates. some metabolic compounds, it will stimulate some genes in the Agrobacterium terminaceae. Those genes are the Vir factor genes, which are stimulated by the phenolic compounds released by the plant roots. The first gene, like the Vir A, which senses the phenolic compounds from the wounded plants, and it induces the expression of other Vir genes. So Vir A, and it will induces Vir G, then Vir G will induces all the genes. Then after stimulation of Vir A, what happens? It passes the information to Vir G. Then Vir G is a constitutively expressed. It will activate all other Vir genes, Then soon after that, what happens? There will be expression of Vir D1 and D2. What they will do? They will bind with the tDNA region on the right border. They will induce the double strand, they will introduce the break in the DNA, and they will synthesize the single stranded tDNA. And Vir D1 induces, Vir D2 holds. Vir D1 is a topoisomerase which helps in the Vir D2 to recognize and cleave the 25 base pairs border sequence, and especially it happens in the right border. Then soon after Vir D2, what it attaches to the 5' end of the T strand, then forming a tDNA complex. This complex is formed. This is called as tDNA complex. Then the another Vir comes, Vir C. It binds to the overdrive region. So okay, another region will be there. Near to this, there will be overdrive region will be there. Binds to the overdrive region and promote the efficiency of the street transitions. Means it will combine Vir D1 and Vir D2 Vir C. It combinedly acts and helps in the synthesis of this tDNA complex. Means your tDNA will come out of from diplasmid by the help of Vir D1, Vir D2 and Vir C. Then Vir E2, which binds to the T strand, which protects, then it has to transfer to this plant cell. It is a plant cell. How it will transfer? The Vir B is the responsible for. Vir B will form a type 4 secretion system. Type 4 secretion system. Like it is like a passage from bacterial cells to plant cell through which your tDNA will complex will move. This is the function of Vir B. Means to create the passage. The passage name is called as type 4 secretion system. Then your Vir D genes will enter into this one. But if tDNA complex will enter into the plant cell, what will happen? The plant will have some mechanisms, will immediately degrade the foreign DNA. To avoid that, we need some protection. The protection was given by Vir E2. Okay? It means it binds the T-tran and protect him from the nucleus attack by the host plant. Then the tDNA will not going to be dissected by the foreign enzyme, like host mechanisms. Then it will go and it will integrate into the plant gene. Then it start expression. That's the importance of. Then Vir E1 stabilizes the Vir E2 actually. Then Vir B told it provides the passage, means it provides some type 4 secretion system, so that from that your tDNA will complex move from the bacterial gene to the plant gene. Now you understood what is tDNA and what are the functions of Vir genes? Any doubts regarding it? Means we are co-culturing both plant cell and bacterial cell, so that when plant cell releases some phenolic compounds and estrous engine, the genes will present, Vir genes is present on the bacterial cell, will do all these actions. Then through the passage is created type 4 secretion system. Then your tDNA will from that bacteria will transfer and into the plant. Then integration will happen. Then there are several markers. One is bacterial selection marker, plant selection marker, same selectable markers, herbicide resistant genes, amplicin resistant genes, same will only come through that selectable markers or scorable markers. We will select which plant is is integrated with the tDNA, which plant is not integrated tDNA. By use of scorable marker or selectable marker, we select those cells and we will do regeneration. Up to this, you understood what is Agrobacterium tuminaceae diplasmid mechanism of gene integration and transformation.
Yes, sir.
Any doubts regarding it? Then, okay, you developed a transgene. Okay, the cell having a transgene, but you should develop a plant. Then only your transgenic plant is created. That's why what we need a plant tissue culture. This is the technique where what is plant tissue? Who given the plant tissue culture technique? Who is the father of plant tissue culture?
Herbert Land.
Yeah, Haberland. Okay, he is the father of plant tissue culture. First, he has used some explants. You can use any explant. Either you can use buds, either you can use roots, either you can use nodal segment, either you can use seed or embryo, whatever it depends on species. Like from first particular species, the buds are very like there is one term called as tissue culture responsiveness, and the protocol for tissue culture for each and every crop it is different. Okay, I have to give some standardization of this. It is not a generalized. Okay, I have to standardize the protocol for each and every crop. Okay, based on the tissue cultural responsiveness or totipotency responsiveness, you have to standardize the medias. Okay, that is another thing. First, there is a first term called as an explant. Means you have to choose any explant based on the species, which for example in tomato buds are very responsive for tissue culture. In mung bean model segment maybe the very responsive. There are different things, okay, but you should remember which is the explant we are using for the uh, tissue culture. Then you have to go for trimming, means you have to cut neatly and you should surface sterilize. Then there are several surface like NOC, NaOCl. There are different surface sterilants were used to sterilize this medium. Then copper chloride. There are many things are used. Then after you have to wash with distilled water. Then you transfer to media. Then you start culturing in the media. You know that media will contains growth hormones, vitamins, minerals, carbohydrates, okay, protein. There are different composition. Majorly used is called as MS media. Okay, MS media. Nowadays the MS media will come as a complete product. However, master mix will come complete mix of all the ingredients. The MS media will come. You just use MS media and you just, for example, MS media plus water, you just make up the volume and you prepare the plates, put your explant and keep in a some incubation chamber. Then after that you have to subculture, means you have to take, for example, you take some part into different test tubes, you have to multiply subculturing. Then you have to screen and you have to grow entire plant. Then you have to go for hardening. Then you have to establish in the field. This is the entire process of a tissue culture. Like there are different forms of tissue culture. If you use embryo, it will become embryo culture. If you use cell, if you cell culture. If you use seed culture. If you use meristem culture. If you use protoplast culture. And everyone have a different. Okay. What is dedifferentiation? What is redifferentiation? What is dedifferentiation? You should know all those terminologies. Okay, I'm not going in detail. Okay, those are very easy things. Then, what is the importance of tissue culture? Because these are not the context of our syllabus, it's a biotechnology. If you know this much is more than enough. Then, what is the role of auxin and cytokinin? These are the basic things will not leave you for the life. Should keep on revising crop physiology and biotech. They are like unwanted friends. You should always remember them even though you're not interested, because our subject is related to this one. You should remember, okay, all your crop physiology and biotechnological things, hormones, importance of hormones. Normally in tissue culture, which hormones are majorly used? Anybody can have a guess?
Cytokinin.
Auxin and cytokinin.
Yeah, auxin and cytokinin. Please remember the ratio. Oxygen induces what, cytokine induces what. If oxygen is more, what will happen? If cytokine will more happen, the balanced ratio will is there, what will happen? Please read it, okay? And read, again I'm telling, read the book Pathfinder by Pranav Kumar, Life Science Part 2. That chapter, biotechnology chapter, is very, very important. Then, what is the importance? We can generate a true to type of clones. What is a true to type clones? What does it mean?
Exact replica.
Yeah, exact replica. And for example, you are using meristem culture. Normally in banana nowadays, actually there is so much demand for micropropagation. Okay, there are so many projects are going. Just if you give sample, if you multiply it, mass multiply it, and you sell it. There are in GK Wiki, the program is going on. You have to just screen, and for the virus, just you can sell. You can generate two to type of clones. Single explant can be multiplied to several thousand. And year-round production. Basically, tissue culture, it happens in the in vitro conditions. It's not having any in-field conditions. You can directly go year-round production, independent of the seasons. Rare and endangered plants can be cloned safely. This I will deal. Okay. While discussing about germplasm, we have a separate class. That's why I'm not going the tissue culture in detail, because in germplasm conservation there are different types, ex situ and in situ. And ex situ conservation, tissue culture is very important. There I will going in detail about these things. What is cryopreservation? What is the different forms are there? We will discuss. Okay. What is the slow growth cultures? We are discussing those things. That's why I'm not discussing much about tissue culture here. And it can long-term germplasm conjunction like cryopreservation under 196 degree in liquid nitrogen. Then, for example, they have some orchids. They are very difficult to propagate in natural conditions. That's why there is a orchid in vitro libraries are created and it protected in the cryopreservation or a slow growth culture. That's why they are used whenever they want. Like there are some wild species are there. They are nobody is cultivating. They are in the stage of critically endangered stage. You just take the some cells, you multiply it, and you store it. That's why the tissue culture have mere important role in the production of transgenic plants, conservation of the germplasms, yeah, true to type of clones, micropropagations, and there are several examples. Some meristem cultures are mainly used to eliminate your viral disease. There are several classical examples are there. Please read it. It's very important for SRB. Then these are the things which I want to discuss about recombinant DNA technology. I hope I have given a brief idea about what is recombinant DNA technology, what are the steps, tools, techniques, and some brief idea about tissue culture. Any doubts regarding recombinant DNA technology? If I teach this much, it's not like that. It is the only portion. Okay, there are, like we have three cores, and still we are unable to learn completely about recombinant DNA technology. There are several things. Okay, we have separate course for just for this one only, recombinant DNA technology, three course. And fundamentals of biotechnology, biotechnological molecular techniques, and genome engineering, separate course. And in molecular techniques for trait integration, 4 to 5 crores course. Uh, we will deal with this actually, these topics, 6 months. It's a very large topic. If you are very much interested, please read T.A. Brown book. And I have taken all these images from T.A. Brown, T.A. Brown book of gene cloning. I have shared na gene cloning. Or you can, if you want more information than this, there is another book called as Primos. Molecular principles of molecular techniques, I think, by Primos. You just type Primos book on biotechnology, it will come blue color, like text page will be there. Please go through that. Okay. And I had Brown is very important. T.A. Brown book is very important for those who are preparing for biotechnological gate exam. Gate is very important. Another book called as Genome. It is also by T.A. Brown, Genome. Or another book for molecular genetics is called as Levin's Genes. How many of you read Levin's Genes? It's a very beautiful book. Those who are preparing for CSI, please prepare this. Levin Genes. Genes by Levins. Their versions will come. 1, 2, 3, 4, 5. There are so many versions. And very in detail. Okay. These are things you should read. Okay. Not like that only BDC you have it because we're all in MSc should come out of from the BDC. Okay? But BDC is important, but other getting knowledge is also very important. Okay, understood what is recombinant DNA technology. There is another class. Okay, there will be another section is there. What is GMOs? Don't worry. How many of you watched my video on GMO? No one. Abhishek, Subracharya, Nitish. No one watched me. Please watch. Okay. Based on what is GMO, what are the regulations of GMO, what are the institutes are there to evaluate the GMO, what is RCGM, what is institutional biosafety committees, what is the role of DBT in transgenics, what is the role of CG, what is the role of Ministry of Climate Change and Environment in the release of transgenics, what is review committee on genetic manipulation, what is GEAC, genetic engineering appraisal committees, and what is the Environmental Protection Act. Okay, what are the rules of 1989? Everything I have discussed in detail in that video. So please go through that video. I covered this chapter in this video. I'm not going to repeat it, that thing. Okay. We'll solve some. If you don't have any question on recombinant DNA technology, hello? I'm audible? Or?
Yes, sir.
Yeah, yeah. Any questions about recombinant DNA technology? Yeah, we'll solve some questions of molecular breeding from 2024 paper. Yesterday I was discussing about what is gene regulatory network. Can you guess the answer? Yesterday we had discussed. No doubts? Okay, sorry, no questions. Yesterday we had discussed what is gene regulatory network, what is hub gene. Okay, otherwise, please go through it. Okay, if somebody should. What is bipartite and directional? Even know? It is nothing but they're having the both expression part and some the part is where your transcription factor will bind and will express. Okay, because gene regulatory network is the network of the gene where some genes are hub genes, some genes will regulate the expression of other gene. For example, they'll this gene will produce some transcription factor, and this transcription factor will go bind this gene, and this gene is expressed. That's what's a bipartite, and it also may be direct. Some will directly affect the other trait, or some will directly affect the expression. They will regulate themselves, and they also regulate other genes. This is the one. That's why both are, and there is a correct explanation. Okay? You read it. Okay, gene. They're very important. The gene. What is gene regulatory network? What is hub genes? I will discuss these things in transcriptomics class. This will comes in transcriptomics. Okay. Then, which of the following DNA marker are predominant in it? At least you should say this answer for this question.
Two, sir. After two.
Yeah. SSR, SNP, and RFL. Because ISSR and RFPD and Scott and SRAP is dominant only. It is just a modification of your RFPDs. Okay, then AFLP and SCAR, AFLP we already know, SCAR also we discussed, they are dominant. Then, which of the following mapping populations show heterogeneous genetic background? Discussed already, except nils, all are having heterogeneous background. Okay? This is what. Then, which of the following marker system is called as selective restriction fragment amplification? Should tell this answer.
AFLP. AFLP.
Yeah, we discussed in detail. Okay, how we are using selection nucleotides. See everything. These are the slides. I didn't change even single slides. All are previous year slides. And previous year before the exam only I have discussed. So many things came in the exam. Then this I have discussed. Mutant line with herbicide tolerance.
Robin.
Robin, we have discussed. Okay. And last year also I discussed about 1987 and 1985 varieties, how they have developed, which mutant they have used, which herbicide. Maybe next year, maybe like for which herbicide this question will maybe a questions. Okay, immediate PR. There's I will discussed. Okay. Then what are the correct about restriction now? You should tell this answer. I also discussed these things while discussing about RFL. How linkage map is created?
3-part test cross.
3-point test score, or you can use any markers, whatever it may be. We need a recombination frequencies. But your restriction map will give recombination frequencies. I told you what is restriction map is the
Option B
example of
and D. Second
ah,
option is
okay.
correct.
I will come. Restriction map is the example of physical map. We discussed now what is physical map, cytological map, linkage map, and the day.
Yes.
Yeah. Then clearly, it's this statement is wrong. Means A wherever the A is there, you just put wrong. Then point mutation can be detected if they are present in the restriction set. Absolutely, what your RFLP is dependent on this. Means B is correct. Both 2 and 4 will be there. B is correct. Then point mutation can be detected easily even if they are located between the target sites. Means between the target. If one restriction point here, another restriction point is. If it is in between, how it can be used? How we can able to do polymorphism test? If this mutation if it is there in the restriction site, based on creating a restriction enzyme, here there will be cutting, here there is no cutting, then you can get polymorphism. But he is telling the mutation is not there in the restriction site, but between the two restrictions. That's why you can't use it. C is wrong. Okay, only B and D will be correct. This option is also wrong. Restriction map measures distance between the sites of cleaved restriction engine. Based on this, we'll based on partial restriction, because this space pair to this space pair cutting is happen. This space pair to this paper cutting is happen. Okay, your gene may be in this distance. Like this, we can create restriction enzyme map by partial enzyme digestion. So one technique is there. How we can map the gene by restriction enzymes by partial digestion. When you treat with the DNA with multiple restriction enzyme, we can able to create a physical map. Okay, understood this point?
Yes.
Yes. The things will actually we have to apply your mind actually. You have to think patiently. Everything will be there and everything will be taught. Nothing from outside of the syllabus or outside of the book. Okay. Just we need to think. You should calculate. Okay. Linkage map is there. Linkage map will comes under like your it is based on recombination frequencies. A restriction map will not give a recombination frequencies. How we can create linkage map? That you should use analytic, like have to be so much think analytically. Then only we'll get the answers. Then we have discussed this reliability of the markers. In previous year also I discussed, but nobody will give attention. Like we are giving attention only for MTAs, marker data association, QTL, genomic selection, genome editing. Okay, they are important, but we also be thorough with the conventional things. Here you can see the marker locus is linked to the target locus. Means your marker is attached to some trait. Marker allele is small m, G and G. The recombination frequency is R. I told 1 minus 2R. Okay. You apply this formula, you will going to get. Okay, your 0.90 will be the answer. Okay, solve it. Okay, it will be easy. These are the things which I already discussed. Then melting temperature. And I told if forward and reverse format we have different melting temperature, how will get amplification? Just directly choose where the option you find forward and reverse which having same temperature, you just tick. Okay, understood?
Yes, sir. Sir, one doubt, sir.
Yeah,
Yes, sir. I can understand, sir, that forward and reverse thing.
yeah,
There is also formula which includes GC content, sir.
yeah. That
We
is
have to.
2a plus t. Sorry, 2a plus t plus 4g plus c. I think this is the formula. 4 into g plus c. 2 into a plus t plus 4 into g plus c. This one formula is there. Okay. It will create melting temperature. Melting temperature plus temperature plus or minus 5 is annealing temperature.
Yes, sir. Yes. Good, sir.
This formula you check either 2 will come here or 4 will come here. I'm getting confused. But this is what the formula is. You just check melting temperature formula 2 into a plus t plus 4 into g plus c. You will get melting temperature. Then for annealing temperature, plus or minus 5 you subtract, you will get annealing temperature. That is one form. But if you not remember, even I am also many times I read, but I am confusing. Either two I have to keep here or I am keeping. But if simple logic is, you should have the annealing temperature always you should be nearly 55 to 58. That is general. Then we discussed, and forward and reverse should have both the same annealing temperature. Then only where annealing will happen, then it will be easy for
And sir,
multiplication.
in forward and reverse, if you calculate like a number of A and T and
Yeah,
G and C, both are same. So we can from that also you can take means still like
yeah,
uh,
that also.
sir.
So in many ways you can think. That is what
Mm,
actually those who will
mm,
think perfectly in the exam. Na, actually, everyone reads same book. Everyone will be in the same part. Those who will like the thinking, you just think in a different way. So that now you told like 8 AGC content are there. Okay, you will get same temperature. That is the one way. And either we can think like this. Okay, we should have both all options having different temperature. Okay, this will be option. Otherwise, you just use the if you know very formula, you can use formula. That's what you should think in a different way. Then only will be reaching the right point.
Yes.
Then then a dominant disease resistant gene in a donor parent is linked with dominant STMS marker with 300 base pair amplicon in a coupling phase. Means if say A and M, they are in coupling phase, dominant allele of the gene and marker allele of the marker. In the marker restricted backward selection, I mean MABB, will be performed to transfer the gene using. If you want, if you want to select this gene, you have to use this marker. Means you want to select this trait, you are using this marker. Which type of this selection?
third option.
You want to select
Foreground
this
selection.
trait, yeah, foreground selection. You just discard your background. Then you check foreground selection based on the absence, foreground selection based on the present. They are saying that they are in the coupling phase. Means capital A is there, capital M is there. Then capital small a, small m. Means they are in coupling. Means A, M, small a, small m. They are in coupling. Then if capital A will be selecting, your M, your sorry, M marker capital M is selected, then capital A of the dominant gene will be selected. Means, and they are saying your dominant band will produce 300 bands. Means if 300 band present, then your dominant gene will be there. So dominant allele of the gene will be there. It should be present or absent.
Present.
Sir, present, sir.
Presence. If it is in repulsion phase, for example, A small m and capital A small. If it is in repulsion phase, what you will expect? Presence or
Uh,
absence?
absence.
Absence. Okay. This is how you have to think. Everything I have discussed. When I was saying this reliability of the selection only. If it is any recombination, there will definitely exchange and they will be like tell I have discussed now like how will selection will happen, what is these phases. That's why you should apply your mind actually. It's not like that if it is like if it asked in this way, I have not thought this question. It's not like that I have not. Okay? You have to think in a very different way. You have to dissect the questions into number of sentences. Okay. Then what is this selection first? They are using marker to select the gene. You just straight away you can cancel the background selection till the foreground selection. They are in coupling phase. They are producing band. Means the band is present, gene will be present. If it is in repulsion phase, the band is absent, gene will be present. Like this, you have to think and you have to answer. Okay. There are so many applied things are asked in this here, and you should be think very in different way to understand the question and you have to answer. Presence of mind is very important in the exam. How much you read is another thing, but the presence of mind is very important. Then, linkage disequilibrium is the non-random association of alleles at a different loci. And in the bracket he mentioned, there finally you are getting linkage disequilibrium in the population. But at gametes, they are in equilibrium. And the statement is correct or wrong?
Wrong, sir.
How it will be wrong? Linkage disequilibrium is non-random association of alleles of the different loci.
Sir, first
The
option is
assortment of
correct, sir. Why
correct
they are
statement
given
is correct,
geometric phase equilibrium
but
for that?
they given gametic phase equilibrium. I told LD may happen by linkage or epistatic effect or some hitchhiking effect. We discussed. Maybe it may happen only for the linkage or population structure. Many ways your linkage disequilibrium happens. Last previous year they are directly asking. Linkage LD will happens only between linked and only between unlinked. Straight away can decide statement is true or right, but they just may modify it in such a way that gametes are in equilibrium, but the population is in disequilibrium. That caused due to random association of alleles. Okay, non-random association of the alleles. That is correct. This non-random association happens maybe due to linkage or due to other factors like evolutionary factors, epistaticity, structure, hitchhiking effect. We discussed. So that's why this statement is true. Then at the same time, the tight linkage between the alleles of different loci results in the high linkage. This statement is correct or not?
Correct, sir, but it can't be the exact
Reason? Yeah,
reason.
because they said that the gametic phase equilibrium. But if the linkage happens, you can't expect that gametes will be in disequilibrium. Equilibrium. If the tight linkage will be there, then definitely you're in the gametes, you're going to get the disequilibrium. That's why A and R both are correct, but reason is not correct explanation for your assertion. Understood? See, simple logic, like simple, just see, simple definition will have how many types of question we can frame. That's why whenever I ask the definition, I'll always force you to tell exact terms. Why it is, if you know the exact terms, the definition, you're not going to confuse if the question is asked in any number of ways. Understood?
Yes, sir.
Then, this is about today's class. During somatic embryogenesis, induced achieved by auxins. Okay, that is correct. The meristematids are the group of meristematic cells that form during shoot regeneration. Okay, then altered expression of multigene families lead to somoclonal variation. Like somoclonal variation, I think many of you know. What is somoclonal variation? During tissue culture, even though you are producing true to type plants, but due to some external conditions or due to multigene families, like altered expression, some exchanges, some other any other epigenetic factor, there will be some mutations or anything that will lead to variation from the parent. That is called as somoclonal variation. During somatic cell regeneration, your genetic variation and that too heritable variation is created. That is called as somoclonal variation. All these things are studied in UG. All are correct. Your statement 1, 2, 3 are correct. Okay. Okay. Then, introduction of powerful leaf traits might outperform the native traits by transferring plants to the synthetic gene. It is called as, okay, I not discussed about cisgenics and intragenesis and transgenesis. We will discuss that point. Okay. Means cisgenesic means we will discuss. First, we will discuss this question and I will discuss that. Have okay. What is cisgenesis means? Cisgenesis and intragenesis means. What is transgenic? For example, BT. From bacteria, the bacterial gene is putting into the background of the cotton plant. Means the gene is from different taxa. Okay, different genera, taxa, whatever, different species itself. But this will have some severe consequences on environmental regulation. Can't approve. What else other can be opted? What else? What option is left for us? To avoid it should not be transgenic, but we need improvement. There is a term techniques called as cisgenesis and intragenesis. Means here in cisgenesis and intragenesis, what will happen? We are bringing the gene not from any species, just between the related species which are easily crossable with this plant. Again, there is a subclassification. Either if you bring only exon from related species, but if you use all regulatory sequence like promoter, terminator from the same species, it's called as intragenesis. Okay. Then, for example, you take promoter and terminator. Means the gene must be flanked by one promoter and one terminator. Sorry, cisgenetics. But the regulatory genes from the related species, like for example, Pusa basmati and non-basmati. If you are bringing some genes from non-basmati to Pusa basmati, only exon, all exon, promoter, terminator, it is called as intragenesis. But if you're only getting from non-basmati to basmati, exon, but rest other like promoter, terminator from the basmati itself, then it is called as cisgenesis. Okay, understood what is cisgenesis and intragenesis? Means you're bringing the gene from related species is cisgenesis or intragenesis. But even though only exon from related species, but immediately flanked by own promoter and own terminator, it is called as cisgenesis. But it is from exon is also different, promoter and terminator, all regulatory part from the related species, it is called as intragenesis. But everything is from different, it is called as transgenesis. Now understood what is cisgenesis, intragenesis, transgenesis?
Yes, sir.
Then you answer this. Cisgenesis and introgenic is they're not introgenic, all they are from same related species. It is native to the plant. But your xeno means outside. It's like transgenic. That's why answer is xenogenic. Means it's non-native. It's a new trait which outperform the native. Means it's non-native. Then xenogenic will be the option. but rest are from native from the plant species. They are intra cultivars only by transferring gene. But in xenogenic, for example, you took taking the gene from bacteria, virus, or fungi, any other. Okay? Understood now? Then in this I will discuss what is base editing. After discussing CRISPR-Cas, I have different class for CRISPR-Cas. I will do. Okay. Then, from which method we can able to create name? Backcrossing, selfing, mutation, chromosome substitution.
Also.
All. This is what. Then this transient technology and history and this human genome sequencing project, I think, and these things all I have discussed in video. Please watch my video on GMO regulations. I discussed in detail. That's why I'm not going again. There are several acts like Environmental Protection Act 1986, Seed Policy 2002. Otherwise, if you're not understood, please text me. I will discuss because already slides are there. It's not thing. Okay. Any doubts apart from this? Tomorrow we will start another thing like genomics. I had discussed, means you should have an idea about what is clone, what is fragment, what is insert, what is cloning, what is library. Okay, libraries are not discussed. Okay, you are creating your cloning. After cloning, you are creating the libraries. Okay, means this plate having the, for example, this colon is having one gene, one fragment, this colon is having another 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 fragment. If you combine all, it will synthesis, if you collect all genome. Means you're fragmenting the genome of a particular plant in several fragments and you're cloning in a one petri plate, like in the bacterial colonies. This is called as library. So you're cloning entire genome, means it's called as genomic library. But if you don't want to do entire genome, you just express this, RNA will come, you done cDNA, then you create cDNA library. This is the expression part of your genome that is called as cDNA library. I have discussed cDNA library and genomic library. These are the different libraries, genomic libraries and cDNA libraries. What is the advantage of genomic library, cDNA library? We should all know about before going to for sequencing. For sequencing, we need idea about vectors, should have idea about libraries, should idea about PCR, should know about what is gel electrophoresis, plotting, all those things. Then only you can understand your sequencing, all those things. From tomorrow, I will start first generation sequencing. Okay. And bye bye, everyone. If you have any doubts, you can ask. Understood? Okay. Classes are going good or we're facing any difficulty? And those who are watching recording, please text me if you are facing any problem. Okay. I know you are all watching recordings because I am getting analytics. Okay. Who will watch how much hours, who is spending how much hour, every information will be recorded. Okay. And please update, like I'm giving again update. Please, what are the new pricing? Please pay that amount, which information was given in the WhatsApp within 15 days. Okay. Any doubts regarding this? No doubts? Shall I close?
Yes. Yes.
Yeah, okay. Then bye. Bye. Good night. Good night, everyone. Bye.
Thank you.